So as mentioned, we're going to talk about the importance of oil, specifically in the context of modern embryo culture using modern incubators and culture approaches. So there's a lot of, I think, misconceptions around oil and a lot of new concepts that are at least being considered a little more in depth recently with modern embryo culture. The impacts of things like viscosity, density, impact of humidity, and some of the differing nomenclature used in commercial products such as heavy, light, paraffin oil, and we'll try to discuss many of these points to add some clarity for embryologists in the lab. So I think most of us are aware oil overlay is a long-standing staple of embryo culture. It's very important. It's been very influential in our ability to culture embryos successfully in the IVF lab. Importantly, it acts as a physical stabilizer for microdrops, so we can get those nice shaped drops that many of us prefer in our culture dishes. But perhaps more importantly, it's a protectant for cell culture as well. And you may or may not be aware, but it provides thermal stability for dishes, especially as you remove them from the incubator for routine observation. It helps provide evaporation and protection from evaporation and the potentially harmful osmolality increase as a result. It helps reduce off-gassing and therefore stabilizes pH. And it can also act as a safety measure or a sink for oil-soluble toxic factors and provide an active protective mechanism. And we'll discuss a little bit about that later. So to expand on that final point, here's just an example study from the mid-1990s where using oil overlay added a layer of protection from oil-soluble toxic factors in the embryo culture media. And this was done from a proficiency testing sample of embryo culture media. And those samples that were cultured with oil overlay were able to successfully grow mouse embryos to the blastocyst stage, while those cultures without oil were not. And so that was an indicator that that oil was drawing some toxic factors from the culture media and protecting those embryos. Additionally, some have used this technique and suggest adding some oil, an open dish of oil, for example, to an incubator to help as a toxic sink, so to speak, to help remove some of those toxins from the gas or the air environment in the incubator. As mentioned, oil can also act as a protectant in terms of temperature and pH, and here's a couple of graphical representations of that. On the left, looking at the presence or absence of oil, influencing temperature of the culture media under that oil. And this was work we had done several years ago at the University of Michigan in varying dish types with varying amounts of media or oil. And you can see across the board, regardless of the dish type, the amount of media or the amount of oil, the presence of oil helped maintain that thermal stability longer than without the presence of oil. Similarly, with pH and off-gassing, you can see data presented here on the right, looking at 500 microliters of media covered with approximately 300 microliters of oil using a real-time pH meter. With the presence of oil, there's a much shallower slope to the graph, showing much slower off-gassing and resulting pH increase with oil overlay as opposed to no oil overlay. As mentioned previously, oil overlay can also reduce harmful media evaporation. This is especially important in modern embryo culture incubators that do not have humidity. And you can see some data here that we published now about 6 years ago or so here at ASRM, looking at microdrops under mineral oil in a standard 35 millimeter dish, and looking at 24-hour intervals over 7 days, the impact of a humidified or a dry incubator environment, even under the presence of mineral oil. You can see in a dry incubator that evaporation occurred as evidenced by an increase in osmolality of the culture media. And there's a significant difference in osmolality after around 72 hours. And so that could have a potential impact on resulting embryo development. So oil overlay can reduce evaporation in these conditions, but it doesn't prevent it, specifically in these modern dry incubators. Time is a factor. This becomes perhaps more important with uninterrupted culture, where there is no media exchange at a 24 or a 72-hour interval. There are several critical variables to consider when looking at the impact of oil in reducing evaporation and osmolality increase. One of those are incubator and the presence or absence of humidification, Another is media, specifically the surface area. And Steve Mullen had done a very nice study looking at some of these factors, published last year in Human Reproduction. Things like media exchange, the starting osmolality of the media, those are all impactful in terms of the impact on the resulting embryos. But for the purpose of today's talk, we'll focus primarily on the oil. The volume of oil, or more specifically, the depth of the oil over the top of the media is an important factor in terms of reducing the amount of evaporation and resulting osmolality increase. And also specifically the type of oil. As we mentioned earlier, there's some confusion with the nomenclature of commercial oils in terms of heavy, light, and paraffin, but there are some differences primarily around the density and the viscosity of the oil. And we'll talk a little bit about that subject in subsequent slides. Importantly, not all oils perform the same. As we heard, there's different nomenclature. As we'll discuss, there are some different properties of oil. And I think most of us know not all oils perform the same. We do quality control. Historically, oil has been one of the most variable components of the culture system. You can see an older study here from 2009 looking at 4 different types of oil, and there was a significant difference in the resulting embryo quality between those 4 oils, suggesting different levels of toxicity between those 4 specific products. There could be various sources of contamination or issues or impurities in the oil, as well as concerns with degradation based on the quality of the starting product or the storage conditions used in the particular laboratories. And we'll discuss some of those points in subsequent slides. Now, we just showed that not all oils perform the same in terms of supporting high rates of embryo development. Going back to that prior topic in terms of evaporation protection and resulting osmolality increase, not all oils perform the same in providing that stability or protection from evaporation. This is some prior work we had presented at ASRM and other meetings looking at the impact of different types of oil on resulting evaporation of media under that oil. And in this particular study, 25 microliter microdrops under around 3.5 milliliters of varying oil in a 35 milliliter dish, cultured for 6 days in a dry incubator with no media exchange. We use Sigma heavy mineral oil and Sigma light mineral oil, since those were readily available commercially and they published the densities. And you can see not much of a difference in density, but there was a significant difference in the amount of evaporation as indicated by the resulting approximate 20 millimolar increase in osmolality of that culture media. We also then compared against 2 clinically commercially available products, a high pure heavy oil and an OV oil, which was marketed as a paraffin oil. And you can see no significant differences there, which again further illustrates the difficulty in determining the true qualities or type of oil based on just name alone. And it's nice to see that at least in the Sigma oil, that there were some numerical indicators to show a heavy versus light oil. But this again points to the differences that some oils may convey on embryo culture. Now, as a final take-home point on this is a more recent paper from 2022 in Human Reproduction, again showing that not all oils perform the same. And this is a nice summary of this very well-written paper. They characterized a number of commercial oils used for embryo culture. And you can see from the tables below, different oils provide different levels of temperature stability, as previously mentioned, prevent off-gassing of CO2 and resulting increase in pH differently based on the type of oil, can differentially impact evaporation and resulting osmolality increase, and can differentially impact resulting embryo development. So this is a very nice study, kind of putting all of that prior data into a current single manuscript assessing currently commercially available clinical mineral oils. So I think that's a good spot to stop and kind of take a step back and ask, in terms of mineral oil, what is mineral oil? Well, it's derived from crude oil. Perhaps that makes it a little less surprising why there is such variability historically in the mineral oils that we've used for embryo culture, considering the starting product. It's a liquid hydrocarbon, usually 15 to 40 carbons long. It's usually a mixture of linear alkanes, cycloalkanes, and aromatic hydrocarbons. And so purification is important, as we'll see, to make sure that you've got high quality starting material. And this is crude oil. There's a fractionating tower. Various products come off as a result. Generally, we're looking to obtain the mineral oils, but again, the purification process is important to make sure you have high quality starting material. Different fractions of oil are available for IVF. As mentioned, the nomenclature is not consistent. And importantly, as we'll see, many oils are mixtures to obtain the desired density or viscosity or physical characteristics. And this can introduce some of that variability that can impact the resulting quality of the oil. One of those primary issues is peroxidation. And so we talked about how many mineral oils are a mixture. You essentially can have unsaturated or saturated components of the mineral oil. Really, it's desirable to have the saturated with no double bonds. The presence of unsaturated double bonds or components in the mineral oil can lead to this peroxidation cascade, and this can compromise embryo development. And this is a nice summary from a few years ago from Klemmer and colleagues, based on largely data from the Atsuki group out of Japan, where they showed that peroxidation of mineral oil can be detrimental to fertilization and embryo development. And there's a series of studies from this group that are very informative. Make sure you look those up in Fertility and Sterility and other places. But they showed that levels of peroxidation in the mineral oil can impact resulting embryo. And subsequent studies have shown that there are factors that influence that level of peroxidation: exposure to light, time, temperature, as well as that saturated or unsaturated status of that beginning product. And that's something that is important for us in the IVF lab in terms of the commercial products that we use to culture our human embryos. More recent publication scientific reports point to another potential issue in oil, and that's the presence of VOCs. As we said earlier, oil can be protective by drawing toxic compounds out of the culture media. Some people put it in the incubator to act as a sink to capture oil-soluble toxic compounds. So perhaps not surprisingly, VOCs can become dissolved in the mineral oil and could then leach into the culture media and negatively impact embryo development. And that's essentially what was shown here in 2017, looking at different types of oil. There was some toxic oil examined. They weren't able to identify any sort of ionic or other differences other than different levels of VOCs in those oils. So that became another endpoint for us to be aware of as embryologists in terms of oil quality and the quality of the product that we're looking at from commercial suppliers to make sure that we're not introducing toxic embryo culture into our laboratories. Now, if there's such a problem with mineral oil because of the quality and the starting material, one may ask, why don't we look at different types of oil? There's certainly different types of oil available, and there are studies that have looked at this. Now, these studies are now over 20 years old, but there have been assessments of silicone oil, looking at bovine embryo development. And again, these oils also have issues. In this particular study from 1995, the presence of zinc or other heavy metals. So there are other types of oil, but they are not without their own issues. And so regardless of the type of oil, you still need to make sure you have good quality starting material and quality control testing to make sure you're not inadvertently introducing toxicity to your culture system. So that leads us to one of the newer products on the market, and that's a synthetic oil. So there are still variables to consider, but there are synthetic oils that can be produced manually rather than from that filtering process from crude oil from the ground. So the properties of this oil can be defined during its production. It can be given desired properties such as viscosity or density rather than mixing oils to get those properties. As a result, this can then reduce the concerns about that harmful peroxidation that we briefly discussed. It can address other contamination issues such as those heavy metals and things that may be present from crude oil pulled out of the ground. And importantly, if this is made in a repeatable fashion under controlled conditions, you can reduce that lot-to-lot variability that most mineral oil lots run into using crude oil as the base component. And so in terms of the embryologist, things that we've benefited from or should be looking for in the oil that we use from embryo culture is improved consistency. That means that the commercial suppliers should be looking for an improved source product. And I think they've gotten much better at that over the years, based on a lot of this data is showing the negative impact it could have if you don't start with a good source product. There's been improved methods of handling and packaging to avoid inadvertent introduction of harmful contaminants. We now know to limit light exposure, control temperature, use uniform packaging such as glass bottles or other issues to avoid introducing toxins, as well as limiting the amount of time that we store or use this oil to reduce that risk. There's also potential treatments that have been done by some commercial manufacturers to further reduce the variability and chance of toxicity, such as washing or even adding special additives to the oil to avoid toxicity concerns. There's been improved testing from labs and suppliers. So there are any number of publications now looking at the improved mouse embryo assay to make it more sensitive specifically to mineral oil or oil and its toxins, using things like extended embryo culture, specific cell stage of mouse embryos used, individual culture, microdrop culture, doing cell counts, differential cell counts to try to determine toxicity. There are sensitive oil-specific bioassays using sperm and other markers, staining of embryos. Morphokinetics, Dean Morbeck has published extensively on that as a more sensitive measure. Now looking at peroxide levels in oils, doing VOC testing, and other sort of molecular endpoints to more thoroughly assess the quality and the consistency of that starting product. So in conclusion, oil is certainly a critical component of the embryo culture system. It's especially important consideration with new dry benchtop incubators because of the concern of evaporation. And one could argue that it's especially important with uninterrupted embryo culture because that would presumably exacerbate that evaporation with no media refreshment over an extended period of time. It's important to ensure oil is not a stressor to the system. We want it to be a protectant in terms of thermal stability, pH, and the other aforementioned methods. But that means having a pure source, thorough refinement, and using the proper type of oil. As we said, there are different types of oil, and there are different types of quality testing performed, which have largely been beneficial for us in the IVF lab. Looking at improved and proper packaging and storing by the manufacturer, we benefit from that in the laboratory, but making sure that we're using and storing under the right conditions, using the right vessels, volumes, looking at things like expiration dates to avoid storing these things for extended periods of time in the laboratory. Importantly, improved quality testing, as we mentioned. And then again, in the laboratory, it's still important for us to make sure we're using product in the appropriate fashion. And I think many products now recommend storing in the refrigerator, making sure that you're having a finite usage so that you're not in and out of that bottle over extended periods of time, where you could have peroxidation occurring in the lab, even though you're starting with a good product. And so that means perhaps storing away product if you're in a small lab rather than using it for months at a time. Thank you very much.